Hello, everyone. I am very pleased to open our next session of the symposium, which will focus on the curious case of Prevotella. In this session, we will have two speakers, Dr. Catherine Lozupone from the University of Colorado and Dr. Cecilia Jimenez from the National Autonomous University of Mexico. Time for Q&A will be right after the two talks as in the previous session. So please make sure to deposit your questions in Zoom using the Q&A button at the bottom of the screen. And please do not deposit questions in Slack. Please note that with the speakers, please note which of the speakers the question is intended for. You can also upvote other questions so we know which ones are in high priority. Each talk will be 30 minutes, and then after both talks, we'll have about 30 minutes for questions for both speakers. So without further ado, I'd like to welcome our speakers to this session. It's my pleasure to introduce our first speaker, Dr. Catherine Lozupone. Dr. Lozupone is an associate professor in the Department of Biomedical Informatics at the University of Colorado. She has been heavily involved in the development of popular computational tools from microbial community analysis, such as UniFroc, that we talked about in day one of the course. Dr. Lozupone's lab focuses on investigating gut microbiome composition and function, as well as its interaction with the systemic immune phenotypes and metabolic comorbidities. She will talk to us today about the immune modulation by Prevotella-rich microbiomes in men who have sex with men and potential implications for transmission of HIV. Dr. Lozupone, please take it away.
Thank you so much for the invitation. Can you hear me okay and see my slides all right?
Yes.
Okay, great. So it's a pleasure to talk to you all today about my work. So work in my lab focuses heavily on HIV-infected populations and the role that the microbiome plays in many aspects of HIV disease pathogenesis. So HIV's main target cell is CD4 positive T cells, and the most large concentration of these types of cells in your body are in the gut. The gut is also an important site of HIV transmission because anal intercourse among men who have sex with men is a main source of mode of transmission. And so the degree to which gut microbes influence the activation state of these CD4 T cells is of interest for transmission because activated CD4 T cells expressing HIV co-receptors are more likely infected by HIV. Once transmission happens, it's been shown that there's a breakdown in intestinal barrier function and alterations in the microbiome as microbial products and parts translocate from gut to blood. They activate CD4 T cells, further enhancing infection. With ART, or antiretroviral treatment, a lot of this is ameliorated, but there are remaining defects in barrier function and systemic immune activation that have been linked with metabolic and other comorbidities in this population. So I started researching this field over a decade ago, and our first paper that we published was in 2013. And in this paper, we used 16S ribosomal RNA targeted sequencing to characterize the fecal microbiome of 4 different cohorts: people with recent HIV infection who had not received any treatment, people with chronic or over a year had been infected with HIV but still had not received any treatment, and then people with suppressed virus on antiretroviral therapy, and then a group of HIV negative controls. And in this paper, we saw pretty striking differences in the microbiome. And so in this picture, each of these is a taxa bar chart. So each color represents a different bacterial family. And one thing that was, I think, pretty visually obvious is that the 3 HIV cohorts on the left have a lot of this pink family. That's the Prevotellaceae. And so Prevotella copri was actually the dominant species in this bar. And they had less of the yellow, than the controls, and the yellow is the Bacteroides family. And this is diverse species within the Bacteroides genus. So when I saw this result, it was not what I expected to see. I've looked at a lot of other microbiomes and inflammatory diseases like inflammatory bowel disease. And, I was expecting more a low diversity microbiome, outgrowth of pathogens that immune dysfunction might bring. And so when I showed this result, my collaborators, Tom Campbell and Brent on the project asked, well, is Prevotella bad? We see so much more Prevotella with HIV. Could this be a problem? And, my initial answer was, I'm surprised by this result because I thought Prevotella was quote unquote good. And there was actually kind of at the time a lot of mixed messaging regarding the potential role of Prevotella in health in the literature. So around the same time we published this paper, there was actually another paper that came out where they showed an expansion of intestinal Prevotella copri in people with new onset rheumatoid arthritis. And in this paper, they showed some evidence that Prevotella copri could be pro-inflammatory. So for instance, in a mouse model, they colonized the mice, they gavaged antibiotic treated mice with either media or Prevotella copri, and they found that Prevotella copri could colonize and expand, and that when they treated these mice with DSS, which is a detergent that affects the gut and can cause inflammation, it's a model of inflammatory bowel disease, they found that the Prevotella gavaged mice lost more weight, and they had a poor endoscopy score. So there was some evidence that Prevotella could quote unquote be bad. We in our hands were trying to take bacteria that we saw increase and decrease with HIV and do immune assays. So we culture we order the most closely related bacteria from a culture collection, we culture them, and we expose human immune cells to them and measure inflammatory markers. And so here I'm showing the ratio of TNF alpha, a pro-inflammatory cytokine, to IL-10, an anti-inflammatory cytokine. And in our assays, so here's Prevotella copri. The color is whether the blood was from an HIV positive or negative individual. That had no effect. But we found that Prevotella copri didn't really, in these assays, act more pro-inflammatory than Bacteroides species that we assayed. But another microbe I'll talk about later caught our eye in this assay, and that was Eubacterium biformis, which has since been renamed to Holdemanella biformis. So despite some evidence that Prevotella could be bad or inflammatory, I was really slow to kind of demonize Prevotella because I had also been involved in work as a postdoc in Jeff Gordon's lab, where we had been looking at the gut microbiome across people in different cultures. And so we looked at individuals from really young to old age in the USA and compared them to individuals from an agrarian cultures, both in rural Malawi and in Venezuelan American Indians. And this is a PCOA plot of unifrac values clustering. Each point represents an individual, and the color represents where they're from. And you can see on PC2 here, we had very clear separation from people from the US from these two agrarian cultures. PC1, I'm not showing here, but that was age. So we have infants here and we have adults here. And this change across cultures was associated with differences in Prevotella and Bacteroides. So this is that same plot, but now I've gradient colored them by their relative abundance of Prevotella. And so you can see individuals in agrarian cultures have a lot more Prevotella and less Bacteroides than individuals in the US. Some individuals in the US pull out towards the agrarian cultures, showing more Prevotella. And there were other papers coming out at the time that showed that this was actually associated with diet in the US. So this is looking at fecal samples from 98 healthy US adults, and they compared the microbiome, whether people were Prevotella rich and Bacteroides poor, or the opposite, and they call these enterotype 1 and enterotype 2. They related this to surveys of what these individuals typically ate, and they showed that individuals with the Prevotella rich, Bacteroides poor enterotype were eating less compounds that would be found in red meat, like amino acids and fats, and more compounds that would be found in more agrarian type diet. So there's really good evidence that, diet might be driving differences in Prevotella and Bacteroides, independent of any kind of pathology that the rheumatoid arthritis papers were suggesting. Since then, there's also been papers that have come out that suggest that in the context of a high fiber diet, that Prevotella is good for your metabolic health. So this is a paper out of the Backhead lab where they fed individuals either barley kernel bread that was high in fiber or white bread that was low in fiber. And they challenged these individuals with glucose and looked at the spike in glucose in their blood. And they found that among certain individuals, those who ate the barley kernel bread had a protection compared to those who had white bread in this spike. And they also showed that these responders who responded well to a fiber diet had a concomitant bloom of Prevotella. So suggesting that Prevotella in the context of a high fiber diet is good for your glucose tolerance. At the same time, though, there were papers coming out saying, well, Prevotella could be bad for heart disease. So again, looking at Prevotella and Bacteroides enterotypes and showing that people in more Prevotella were making more of a harmful metabolite for atherosclerosis called TMAO from components of a high-fat diet. So when trying to make sense of this, I kind of was thinking, it's not that Prevotella is good or bad. It depends on your diet. So like if you're eating a high fiber diet, Prevotella is beneficial. But if if you have a Prevotella rich microbiome and you're eating a high fat diet, it's could be detrimental. And there was actually similar things on the other end of the spectrum. Prevotella and Bacteroides sort of trade off. And there were papers in mice showing that administration of certain Bacteroides uniformis, in the context of a high-fat diet, ameliorated metabolic and immune dysfunction that high-fat diet produced. But these beneficial effects of Bacteroides, again, only occurred in mice fed a high-fat diet, not in mice fed the standard chow. So context-specific interactions of Bacteroides and Prevotella with health, with diet. So what I found interesting was in this other paper that also found Prevotella-rich Bacteroides poor microbiomes in HIV-infected individuals, they related this to diet. And they found that in their HIV-negative controls, there was a very nice correlation between the relative abundance of Bacteroides and the number of servings of red meat per week, suggesting that a healthy person is suggesting for beneficial bacteria in a diet-dependent manner. And this relationship was completely obliterated in their HIV-infected subjects. So that association between diet and the microbiome that helps you process that diet was disturbed. And so based on all of these papers at the time, I kind of came up with a hypothesis that, there's not one healthy microbiota. You want Prevotella if you're eating fiber, you want Bacteroides if you're eating a Western diet. And I thought maybe the reason this gets messed up in HIV is that CD4 T cells have been shown to interact and select for some Bacteroides, and these are targeted by HIV. And so you default to a Prevotella-rich microbiome. So we looked further into this hypothesis, and we recently published a paper on this. So I'm not going to show all the results of it, except to say that, unfortunately, we are not actually finding very strong support of this hypothesis. We did find one amplicon sequence variant, and we had a few that mapped to Prevotella, but one of them predicted metabolic disease in this population, but it was beneficial. So it's more consistent with the paper out of the Backhead lab saying Prevotella could be beneficial for your metabolic health, and not supportive of our kind of, in this population, it won't be. So anyway, we did find a lot of other microbes associated with barrier function, which was associated with poor metabolic health. So there were other microbially mediated mechanisms supported in our work. So we could have maybe guessed that our hypothesis would fail, because very early in the study, we realized our hypothesis was flawed. And it was flawed because we made an error that I think a lot of microbiome papers do. And that was, we did not select an appropriate negative control population in our study. So, so we have, since that initial paper in 2013, a few years later, we published another paper that really provides better information on how the microbiome differs with HIV. Rather than looking at just a couple dozen individuals, now we're looking at 217 individuals. And in this table, I'm showing you the degree to which the individuals in our HIV negative, HIV positive, ART naive, and HIV positive ART treated cohorts distributed again across 3 different groups. So we have women, and then among men, we have men who have sex with men and men who have sex with women. So as I mentioned, anal intercourse among men who have sex with men is a major mode of transmission of HIV, and it is very much in the populations in Colorado we're looking at. So if you look here, among our HIV positive cohort, the vast majority of them are men who have sex with men, even more so in our untreated cohort. And so in our prior study, all of our controls were just selected matching for things like age and gender, but not sexual behavior. But in our work moving forward, we began to recruit from a cohort of individuals of men who have sex with men who were at high risk of acquiring HIV. So all these individuals were actually recruited for a vaccine trial. They were having sex with many other partners, unprotected, so high risk activities. And so when we now had a control population with some MSM, men who have sex with women and women, it painted a different picture. So here's another PCOA plot. And here the points are colored by female in pink, men who have sex with women in orange, and men who have sex with men in green. And then the shape is whether they have HIV or not. So circles are HIV negative, and HIV positive, treated and untreated, respectively, are the squares and the triangles. And you can see we have clustering by sexual orientation and gender rather than HIV infection status. And so, and when we looked, so now here's a plot showing, this is in this, it's a taxa bar chart, but it's they're ordered in the same order as PC1 here. You can see this very striking pattern. The pink is the Bacteroides, the yellow is the Prevotella. As you move from the side where you have the non-MSM to the MSM, you get a loss of Bacteroides and a gain in Prevotella. So this opened up all these questions, right? Like we thought this was HIV driven, and we had all these hypotheses. It was driven because defects in CD4 T cells were disturbing interactions with Bacteroides. So this is not true. So we had to then step back and ask, what is even the difference in the microbiome in HIV when we're controlling properly? And then also, why do the high-risk MSM have this different microbiome? And when we were recruiting this cohort, we actually, had some information on sexual behaviors. We've started asking more questions on that than we used to. But what we tried the best with the data we had. So for instance, this is a figure from that paper where it's the same plot, but these are indicating whether men and women are engaging in receptive anal intercourse, which you would think would be one reason they might have a different microbiome, and it didn't explain it. So we had women who engaged in RAI who still clustered with other women, and we had men who never engaged in the receptive part of anal intercourse, and but were having sex with other men, and they were over here. So at this point, I cannot tell you why the microbiome is different in this population. I can just tell you it is. And so really then, because our biggest microbiome differences were in these high-risk MSM, we just had new questions about, now we had 2 questions about both the HIV-associated microbiome and its effects on immunity, and also the high-risk MSM microbiome and its effects on immunity, because this is a very important population because they are at high risk of getting HIV. And so one of the first things we wanted to do is really just better understand, are there immune differences in our high-risk MSM populations? And we found that there were, and there were some in actually some very important immune populations for HIV disease pathogenesis and HIV acquisition and control. And so in one paper, like we looked at the blood immune markers, and we were specifically looking at ones that people in the HIV world really care about. So for instance, CD8 T cells expressing these activation markers are important markers of morbidity, disease progression, comorbidity in people with HIV. And it has been shown in many papers, it's elevated in HIV positive people, but it was actually also already elevated in our high risk men who have sex with men compared to our non-MSM. And then this is another interesting immune type in HIV, because CD4 T cells are the target of HIV, and CD103 are the percent of those that are, that's a mucosal homing marker. So these are moving, you know, they're moving either to or from the gut, the lungs, something like that. And MSM had more of these than non-MSM. And HIV positive didn't, which actually makes sense because HIV kills these cells. But the fact that they're higher, these target cells of HIV and MSM, is interesting. We also were interested, because transmission is really important in this population, on whether the microbiome could influence the expression of the HIV co-receptor CCR5 on CD4 T cells. This is required for HIV to infect T cells. And again, if it's activated, these are activation markers, then these cells are going to be even more likely to be infected. And we found, using biopsy samples and looking at rectal biopsy samples, that our high-risk MSM actually had a higher proportion of their T cells were expressing these markers that would make them readily infectable by HIV. And then we also, in a more untargeted search, where we actually used technology called CyTOF to screen many immune populations, we found this very interesting population called mate cells was depleted in MSM, whether they have HIV or whether they don't have HIV, compared to our non-MSM controls. Mates are interesting because they're enriched in tissues. They're important in mucosal barrier integrity, maintenance, and repair. And they're known already to interact highly with the microbiome. In fact, they respond specifically to bacterially derived riboflavin synthesis intermediates. And previous studies have reported a loss of mates with HIV and made the same mistake we made. So this has everything to do with HIV, like, you know, like influencing the immune system. But having this important control population shows it doesn't actually have anything to do with HIV. But this is important in this population because mate cells actually can respond to and suppress HIV. So again, a loss of this in high-risk populations is potentially going to promote the spread of HIV. So the next thing we want to do in these studies, this is all, we found differences in the immune system, we found differences in the microbiome. Are these related to each other? And so to do this, I collaborate with an immunologist, Brent Palmer, to do a lot of lab work. And a lot of the work I'll show you today was done by two different postdocs in his lab, Sam Lee and Preston Neff. And so there's two different types of assays we'll do. One is we take either cultured bacteria or whole bacterial communities from feces, and we use them to stimulate immune cells. I already showed on an earlier slide a result with stimulating peripheral blood mononuclear cells. We'll also get gut cells from leftover surgical surgery tissues. And then we also use notobiotic mice. So rather than look at individual bacteria, like I showed you on that earlier slide with Prevotella and the Bacteroides having similar pro-inflammatory signals, here we are actually taking like the whole feces. And so we're taking a fecal sample, we're using histodense column to get out the whole intact bacterial cells. We check how good our prep is with flow cytometry, and then we also sequence it, and we can show that our isolated bacteria have similar compositions to the whole stool, and we're able to reproduce, bacterial preps that are enriched in Prevotella versus Bacteroides to use in these assays. And so, like, we take the fecal microbiota, expose them to PBMC, and look at flow cytometry of these cell populations. One limitation of this are these microbes are dead. It's not like working in a mouse model, but it's sort of telling you, like, the sum of their parts, how those components of their cells might be influencing the immune system. And so one of the things that we did, remember we had, and we published this in Gut Microbes a couple years ago, one of the things that we were interested in were these CCR5, because it's an HIV co-receptor. And what we found in these assays was that, so here we have non-MSM, and then we have men who have sex with men who are HIV negative, and then men who have sex with men who are HIV positive, either are treated or not. And all three of these MSM populations, regardless of HIV infection status, had elevated stimulation of CCR5 on their CD4 T cells, suggesting that what we had observed just looking at biopsy, that these populations were higher, and that the microbiome as a whole was actually a potential driver of this. It supports that it is a driver of it. We also were able to do infection assays. So very similar, you take the fecal microbiota, you expose immune cells to them. Now we're actually doing immune cells from the gut. A surgeon, Martin McCarter, he does gut resection surgeries, usually for tumor removal. He gives us a healthy tissue. We get out the immune cells. We stimulate them with the fecal microbiota, but then we actually also try to infect them with HIV. And what we found here is that the cells, the gut immune cells, T cells that were pre-stimulated with the fecal microbiota of MSM, whether they were HIV negative or positive, were more readily infected by HIV compared to the men who have sex with women controls here, or here's media. So that told us that the microbiome as a whole was having, really important drivers potentially for transmission. And so next, we used the same data that I showed you, where we correlated, where we showed elevated CCR5 positive cells and MSM, and we looked to see which microbes were correlating with this. And we didn't find Prevotella, which was our most dominant microbial difference. Instead, we found Holdemanella biformis as our top positively correlated microbe. And if you remember from the slide, that was the one that we had already tested in vitro and had a really pro-inflammatory cytokine profile. And actually, the only negatively correlated one was a Bacteroides, an ASV mapping to Bacteroides. So it could be the lack of Bacteroides that's also contributing. And I'm not going to go into too many details here, but we recently published another paper where we, took out a couple of strains of, actually cultured some strains of Holdemanella out of patient samples. We compared their ability to induce HIV co-receptors to Bacteroides uniformis, and one strain was higher. They were all trending. And then actually that one strain also had a particularly pro-inflammatory profile, cytokine profile. So we compared their genomes, and they had differences in these capsular materials that could be responsible for these differences in immune modulation. So we're continuing to research, how and why certain microbes could be driving these important phenotypes we see. We also used gnotobiotic mouse models. So again, we took these three different patient populations, and we looked at this, and we didn't get perfect colonization, but we did get an increase in immune activation with the MSM regardless of HIV status. And we put Holdemanella in the mice, that same species, and we could see that actually would increase it, and it correlated. So we were still getting evidence of the same microbe. So what about Prevotella? Prevotella didn't colonize the mice. And so here's the profile, and you can see Prevotella in gray. It was in the donors and not the recipients. This was surprising because prior studies have shown Prevotella does. We took Prevotella copri, it did monocolonize. And interestingly, the Prevotella from our HIV negative individuals did colonize, suggesting some sort of strain level variation. And indeed, when we actually look at the variation in MSM, there's a few different strains. These are different ASVs of Prevotella. Most go up, one goes down. And so I think that Prevotella is showing some strain level variation in this population, which would be very consistent with what we've seen in comparing Prevotella from people with westernized lifestyles generally to non-westernized. There's great differences there. It's actually a very complex complex of different strains with not a strong core genome. And there's more strains in the developing world. It's kind of a paucity of strains in the US. And this has been associated with differences in their genomes in terms of carbohydrate breakdown. We are in the process of getting shotgun metagenomic data so we can see what this looks like with our MSM populations. So in summary, populations of people living with HIV who are MSM and high-risk MSM tend to have Prevotella-rich Bacteroides poor microbiomes. We hypothesize this might influence their metabolic health because of a diet microbiome mismatch, but we didn't find evidence of that. We did find evidence that this is more of an MSM phenotype, and this Prevotella-rich Bacteroides population microbiome is correlating with undesirable immune phenotypes. But immune in vitro assays and mouse models are really pointing to some other microbes that correlate with Prevotella, not Prevotella itself, and driving it. And so we've really honestly been looking more at other things, although we are going to look at that strain level variation in Prevotella now, because I feel like that could be actually obscuring the detection of any role if we're just binning all the Prevotella when they shouldn't be binned. So with that, I will stop and thank the people in my lab who did the work. A lot of the analyses were done by Abigail and Janet and Nicole, and also all of my HIV collaborators, our study participants, and the NIH. Thank you.